Hi everyone, I'm Becca Schulte,
I'm Elizabeth Lynch.
And I'm Lucas Bennett.
and for our research project, we took a parasitic survey of saltwater catfish from the Tampa Bay, Florida, and we specifically looked at Arius felis and Bagre marinus species. Arius felis is also known as the hardhead catfish, which belongs to family Aridae, and this family is going to consist of 26 different genuses and over 143 species, which is going to include our second species, which is Bagre marinus, or the gaff-top sail catfish. These species are going to share a native range around the United States, the Caribbean, and Mexico, with a distribution throughout the Atlantic and Gulf Coast as brackish estuaries and along the coastal waters. These species of catfish are going to be considered opportunistic consumers, spending the majority of their time hunting on the muddy bottoms and scavenging for pretty much anything they can fit into their mouths. We're typically going to see that the juvenile hardheads are going to eat smaller crustaceans such as shrimp or mollusks, and the fully mature adults will feed on larger fish. But like I just said, we're primarily going to see them as opportunistic consumers. So they're going to be a very involved species within these estuaries. So the hardhead catfish are preferred diet species over other estuary species, and it's going to be assumed that in this case with Tampa Bay, that this hardhead species is going to be preferred by the bull sharks we see. So it's going to be thought that these catfish are going to be acting as some sort of intermediate host for parasitic infections found in the bull shark species. The family Aridae has had previous parasitic surveys done showing a metazoan parasite community along with another common protozoan parasite that we refer to as the ick. And we're going to see that along the gills and the skin. However, the past surveys done with the Aridae family cannot be directly related to what we're going to find in our results, just because the species we examined have little to no parasitic research or surveys done in this area around Tampa. So we're going to move on a little into the methodology. So our survey is going to start with two different location dates. So the specimens were collected in the summer and spring using a 100-foot gill net along the Alafia River in Hillsborough County. The summer collection took place from June through August of 2021, catching a total of 28 catfish, 26 being the A. felis species and 2 being the B. marinus species. The spring collection took place in February of 2022, and the catch consisted of 10 total A. felis species. And so these two sample data collections allowed us to have a pretty normal sample size of 38 total specimens. And Lucas is going to go into a little more depth about the methodology.
Alrighty. So yeah, I'm going to talk to you guys more about our methods and specifically our dissections and how we went through and found these parasites. So we went and examined all 38 specimens internally and externally for parasitic infections, looking for any endoparasites and ectoparasites. We used dissection microscopes in order to go through all of the internal organs as well as any connective tissues. And we also looked over the gill filaments after removing the operculums of each one of our specimens to possibly find any monogenetic parasitic infections in the gills. Unfortunately, we did not find any monogenes in their gills, but we went through their other internal organs and we were able to find 2 parasite species, one being a larval cestode and an acanthocephalus species, which is a thorny-headed worm. Further with identification after we found these parasites in the organs, and also some of the organs that we went through inside consisted of the stomach contents, small and large intestine, gallbladder, spleen, heart, and kidneys. We also went through their livers too, picking them apart with dissection tweezers as well as looking under the dissection microscope to make sure that we weren't missing any parasites. And as you can see in our pictures, we have the gallbladder in that top left middle column, as well as a cystic duct, which is the left part of the gallbladder, which is where most of these larval cestodes were being found. The middle picture in the middle column is in the top middle is going to be the gallbladder itself and the larval cestodes found inside of the fluids within the gallbladder. And then in that right picture in the top middle is going to be the small intestine of the A. felis that we dissected, or those hard heads that we dissected, and we found those acanthocephala sitting in the digestive tract, specifically in the small and large intestine. We did find some in the stomach as well, but mostly in the small and large intestine throughout. Once we found these, we preserved them in 70% ethanol. Some put 95% ethanol for possible PCR analysis, but the 70% ethanol was used for staining. We stained with acetocarmine and also destained with a 5% hydrochloric acid solution, as well as prepping them with xylene and mounting them on slides with a xylene mounting medium. And we did that in order to further identify these, or try to further identify these two parasites using light microscopy. And out of 37 of the 38 catfish that we went through, we did find endoparasitic infection, either for the larval cestodes or both larval cestodes and acanthocephala. Prevalence, abundance, and mean intensity were calculated for both acanthocephala and the larval cestode species within A. felis and also B. marinus. And with our stats, we ended up comparing the mean abundance of larval cestode infection with the standard length to the catfish to see if there was any correlation there. And we also ran Mann-Whitney-U test to see if there was any significant difference between gender and the parasitic abundance for the larval cestodes. And so we did not find any significant difference for the Mann-Whitney-U test when comparing genders and larval cestode abundance. And we also did not find any correlation between the standard length of the catfish and larval cestode abundance when looking at these specifically. And Liz is going to talk to you guys a little bit more about the rest of our paper.
So just to discuss more about like our results, our A. felis and B. marinus like Lucas said, they both showed high prevalence and mean abundance, as well as mean intensity of the larval cestodes within the gallbladder. And these larval cestodes were found without, not just in the gallbladder, they were found in the liver and the kidney as well, but they were found in much lower numbers. As well as the acanthocephala. The acanthocephala was found in the A. felis in both of the collection dates, and they were found within the intestines. And we couldn't really say with the B. marinus species whether or not these parasites were in these species or not, due to the limited number of these species we had. We also found no correlation between the parasite abundance and the standard length due to the Pearson R correlation test. And then, as well as according to our Mann-Whitney-U test, the larval cestode abundance and its gender, as the U statistical score was higher than the critical U value, it showed that there was no statistical difference between gender and the parasitic infections. There could possibly be a seasonal difference between the specimens and their parasitic prevalence, abundance, and mean intensity. Due to the abundance of the parasites, it was found much higher in the spring collection compared to the summer, although more studies are needed to determine any statistical significance between the seasons and a parasitic infection. The small sample size we had from the spring collection made it hard to determine whether or not there was a real relationship there. The larval cestodes have yet to be identified and described by the scientific community. However, them being with such high prevalence in these preferred species, we can assume that the larval cestodes are easily transmittable to higher predators such as the bull sharks, and there were bull shark species found in Tampa Bay. So the possible identification of these larval cestodes will allow for a completing of its life cycle. Another research group in our class was doing a parasitic study on bull sharks in the same area, in the Tampa Bay area. However, due to time restraints, we were not able to make the connection between the larval cestodes and the parasites found in the bull shark. And basically, to conclude everything up with our research, this parasitic survey of A. felis and B. marinus found new host records, which wasn't too surprising given the lack of previous studies done on the endo and ectoparasites found among catfish in Florida. The gallbladder and the digestive tract were the main points of infection, finding the larval cestodes in high abundance of the gallbladder and the acanthocephalos in the digestive tract. The mean abundance of the larval cestodes from the summer collection was approximately 131.65 and rose to about 245.4 in the spring collection. Mean intensity saw the same trend, slightly increasing from the summer to the spring collection from 136.92 to 245.4. Again, there was no statistical correlation found with the abundance of the larval cestodes and standard lengths, and there was no statistical difference between larval cestode abundance and gender.
And with that, we'd just like to acknowledge Dr. Langford and his summer research group for being the ones to collect these specimens for us in order for us to conduct this research. And we have our IACUP reference too, as well as our acknowledgements. We also want to thank Dr. Langford for all of his guidance throughout our research this semester and last semester, as well as the biology department of Florida Southern College for allowing us to conduct this research on campus, as well as all their support throughout our entire research. So